Okay, hi students, can you all hear me? Yay! Welcome. I'm so glad I finally got it to load. It was not loading for whatever reason and I finally refreshed it and it's working. Yay! I'm so happy to see a lot of you from BTech. Yay, I'm so glad you guys made it. All right, awesome. So let's go ahead and get started. So I'm Miss Aguilar. I am an AP Bio teacher in Los Angeles, California. And we're going to review the FRQs for 2016. So what I'm going to ask you guys to do in just a few moments is to go ahead and pull up the FRQs yourself because I'm going to be going through some slides with you, but you also need the FRQ questions to go along with the slides, okay? So it's good to see all of you guys, lots of highs and hellos. So let's go ahead and I'm going to share my screen with you so that we can get started here. Okay, so just so you guys know, they have a weekly stream calendar for Fiveable. So if you're interested in joining Fiveable for any of your other AP classes, you can also do that. So this is the schedule here that you see. For biology specifically, they host streams on Wednesdays at 8 p.m. as well. And that's Eastern time, I believe. Okay, so let's move on. If you want to follow Fiveable, it's at Think Fiveable. So like I said, I'm Miss Aguilar. I am an AP Bio teacher in Los Angeles. And let's go ahead and get started. So let me move this here. Okay. Just getting set up here. All right. So right now, as of right now, what we know for the current structure of the AP Bio exam, you are going to have 25 minutes to respond to the first question, okay? And then 5 minutes to upload your response. So you can take a picture, say you draw a graph, or you have to do some kind of data analysis. You have 5 minutes to upload your picture after your response. After uploading your response to question 1, you're going to have about 15 minutes to upload and respond to question 2, with an additional 5 minutes to upload your response for question 2. So once your response for question 1 has been submitted, you cannot go back into it. So just keep that in mind. They have told us through the College Board that you're going to have question 1 is going to be 65% of the exam, and question 2 is 35% of the exam. Okay, so let's move on to the next slide. The questions are going to be about 2 major topics. So the first thing is interpreting and evaluating experimental results. Question 1 presents students with an authentic scenario accompanied by data in a table or a graph. This question assesses students' ability to do the following. So you need to know how to describe and explain biological concepts, processes, models, and so on. You need to be able to identify and justify experimental design procedures, analyze data, and make and justify predictions. So you need to be familiar with experimental setups. So for example, knowing what a control is, knowing the independent variables, etc. That's all going to help you when you're doing question 1. So we're going to go over some of that stuff in a few minutes just to get you more familiar with all of it. For question 2, you are going to be given some kind of scenario describing a biological phenomenon with a disruption. So basically, understanding a biological concept, explaining that concept, and then predicting the cause or effect of a change to that. So that sounds a lot to me like feedback loops and mechanisms. So if you haven't covered that, it would be a good idea to probably look into feedback loops just as a reminder, refresher for you. All right, so let's continue. So some of the FRQ tips. This following strategies for answering FRQs will help you on exam day. This is all from the College Board, and I just edited the most important key points to try to help you guys. So the first thing that you need to know is that the first question is going to be divided into parts, such as part A, B, C, and D. And you will be writing a response for each different part. You can get credit for each part that is that you actually attempt. So you should attempt to solve all of the questions. But obviously, if you don't know something for, say, part A, you can skip part A and you can go to B and C and D and still get points for that. So that's something really important to realize. And the other thing is you can organize your answers as clearly as possible. So you might want to label all of your answers. So starting with A, and you write your response, and then label section B, and so on, so that you have organized work, okay? The last thing is to be sure to clearly and correctly label all the graphs and diagrams that you make. So read the question carefully, as this could include a graph title, the X and Y axis labels, and units, and so on. Okay, are there any questions about the FRQ tips so far, or any of the new changes to the AP Bio guidelines? All right, so I don't see any questions, so I'm going to go ahead and move forward. So what I need you guys to do right now is to go ahead and search on Google. I want you to search this, which is the 2016 FRQs for AP Bio, so you can follow along. We're going to begin with question 1. All right, give me a thumbs up if you are ready to go. Yay! Okay, so what you look up is 2016 FRQs for AP Bio, because it's going to come up with the questions for the FRQs that we're looking at. All right, I'm seeing lots of thumbs up, so let's go ahead and begin. We're going to start off with question 1, so everyone get to question 1 on your screen, and let's move on to the next thing. All right, so some steps to basically answering FRQs. You want to look at read the question, first of all. You want to break it down. And then for step 2, you're going to kind of summarize the question in your own words. And part 3 is basically staying organized, so making sure we're answering all the sections correctly. All right, so let's go ahead and read over part 1, question 1 together. So I'm going to pull it up on my screen as well. All right, so the first part of the prompt is just the introduction, so we're skipping that. But we can see that the first thing they give us, well, first of all, this is question 1 and 2 are 22 minutes each, and they're worth 10 points. So the first question that you get on this year's AP test is obviously worth 65%. So you are going to want to try to attempt that question over the other question more closely, okay? But let's read the question together. We are seeing a figure 1 for all of the Long Island data, but we're going to skip over that just for now. I'm going to go ahead and read the question for you guys, okay? So it says, leucine aminopeptidases, LAPs, are found in all living organisms and have been associated with the response of the marine mussel Mytilus edulis to changes in salinity. Okay, so I'm going to just stop right there. Obviously, you don't need to know all of this stuff. Leucine aminopeptidases, that's a really long word or words that we don't really necessarily need. So what we can say is, oh, this word ends with ASE. So does anyone remember when words end with ASE? what they are signifying. Ah, enzymes, yay! Okay, good. It's good that you guys know that. So, moving on. So we know that this is an enzyme, okay, and it's found in marine mussels. So if you look at this little picture that I have in the corner of my slides, you can see all these mussels. They like to chill on the coastlines. They like to chill on rocks, and they, when the tide rolls in, they gain some of that salt water. They gain their nutrients from the ocean, okay? So that's just for some people who may not be like in the East Coast or West Coast like we are. Okay, so let's continue with the question. So we know so far we have an enzyme that's found in marine mussels. All right. And it helps with changes in salinity. So salinity has to do with the salt water concentration. All right, so LAPs are enzymes that remove N-terminal amino acids from proteins and release the free amino acids into the cytosol. To investigate the evolution of LAPs in the wild populations of M. edulis, again, we don't need that word, we can just skip right over it. Basically, in populations of mussels, researchers sampled adult mussels from several different locations along a part of the northeast coast of the United States. Okay. In figure 1, as shown in figure 1, the researchers then determined the percent of individuals possessing a particular LAP allele, LAP94, in mussels for each sample size, each sample site. All right, so now it's asking us for A, on the axis provided, construct an appropriately labeled bar diagram to illustrate the observed frequencies of the LAP allele in study populations. So going back to what I was saying before, we want to break down and summarize the question. So in summary, we see mussels are marine organisms that respond to changes in saltwater concentrations by using the LAP enzyme. LAP enzymes add amino acids to the cytosol, which is the fluid found inside of the cell. Okay, now we're going to answer the questions by staying organized. So starting with question A, like I said before, if you didn't understand question A, you could move on to the next question. But because we're doing this as a review, obviously, we want to make sure that we are answering every question. So that's relevant, at least. All right, so on the axis provided, construct a bar diagram. So it's telling us we need a bar graph. That's good because now it's already part of our job done. So go up, scroll up to the table. You can see that we have a site and the LAP frequency. So which one do you guys think is going to go on the x-axis if we're graphing this? Yes, that is correct, Raul. Yay, thank you guys. The site is going on the x-axis. So if you have a piece of paper and you can kind of just do a sketch of the graph, that would be great. This is question 1 where we have to look up the questions for the 2016 FRQs. Okay. The site goes on the x-axis, and then what do you guys think is going to go on the y-axis? Yes, the LAP frequency. Okay, and then we said we're going to be doing a bar graph, so all we would have to do is write out all of the sites on the bottom. You would space them out evenly, and then we are going to graph the LAP frequency on the y-axis. So what do you guys think would be a good number to go by on our graphs? Make a suggestion. Yeah, 5 could work for sure. I would probably go with 10. 10s is just, I mean, it's just easier. It'll make your graph a little quicker. So I personally went by 10s. If you went by 5s, that is also 100% okay. One thing that is important to remember, and a lot of students, not usually at the AP level, but in the past, like with just regular bio students, sometimes students struggle with remembering the scale. So you need to have the correct scale, as in if you go by 5s, you need to go by 5s all the way up the y-axis. If you go by 10s, you're going to continue to do 10s all the way up the y-axis. All right, so perfect. You guys seem like you know what you're doing, so let's move on to the next part. So we should have graphed our information, and then we're going to move on to the next stuff. So part B, so label part B on your paper. Based on the data, describe the most likely effect of salinity on the frequency of LAP94 allele in marine mussel populations in Long Island Sound. Then it asks us to predict the likely LAP allele frequency at a sampling site between site 1 and 2. All right, so now what it's asking is, what is this enzyme basically doing? What effect is it going to have on the LAP allele? Okay, so let's take a look. So as the salinity increases, what do we notice is happening with the LAP allele frequency? Yes, that's right. It does increase over time. Over the sites. Yay. Going back to Lay's question, can we go by 1 on the x-axis? Yes, we have to because we're labeling the sites on the x-axis. Okay? And yes, you can go by 10s on the y-axis. Okay, so good. So we're seeing an increase. So going back to part B, as salinity increases, the LAP allele frequency increases, and that is actually describing the most likely effect of salinity on the frequency of the LAP allele. Okay, if you said the opposite, as salinity decreases, the LAP allele frequency decreases, that's also correct. So that's perfect. What about a prediction for the sites? So we want to know, what is the likelihood of having the allele frequency between sites 1 and 2? What do you guys think? Yes. Good. I'm seeing lots of right answers so far. Basically, you can put anything between 13 and 16%, or you can put a value. So like if you put 14, 15, 14.5, all of those would be correct for part B and your prediction. Okay? Excellent job, guys. So let's move on to part C. Describe the most likely effect of LAP94 activity on osmolarity of the cytosol. Okay, so we're looking at how is the LAP enzyme actually affecting the cytosol, which is found inside of the cell. What is actually being added to, what did we say was being added to the cytosol? Yes, so we have the amino acids being added to the cytosol. So basically, we're going to say does that increase the solute concentration in the cytosol or decrease the solute concentration in the cytosol? Yes, it increases the concentration in the cytosol, okay, because we have a bunch of amino acids, that's lots of solutes in the cytosol. So what does that mean when we go back to osmosis? Where is water most likely going to move if there's a high concentration of amino acids inside of the cell? Water will move inside of the cell. Yes, water is going to move from where it's at a high concentration to low concentration. And yes, if you're using the terms hypotonic and so on, that's also good. All right, so let's answer that question. So the most likely effect on the LAP enzyme is that it increases the solute concentration of the cytosol. Okay, that's describing the effect of the LAP activity. Now we're going to describe the function of LAP in maintaining water balance. So basically, if water is going into the cell, then we're preventing water from being lost to the environment. Okay, so that would be the second point that you would get. All right. Give me a thumbs up if you are keeping up with us okay. So osmolarity. Okay, so you really don't need to know that term in particular. Like it's basically just asking about what is the concentration inside and the concentration outside of a cell. Whenever we have a high concentration of solutes, that has to deal with osmolarity because water is going to move in by osmosis. Okay, so I hope that helps. All right, I'm gonna give you guys a few more, like maybe another 30 seconds to answer this question. Okay, if you are caught up and ready to move on, I would suggest trying to answer part D on your own, and then we'll go over the answer right now. All right, for Tim, so high concentration means high yes, osmolarity. Osmolarity means there's a high concentration of solutes inside of the solution that we're looking at. So I'm being asked the question, could you, let's see, it says, what is the function of the LAP in maintaining water balance? So the LAP enzyme is basically adding a bunch of enzymes. It's adding a bunch of amino acids into the cytosol, which is causing the concentration in the cytosol of the cells to increase. Okay, so if the concentration is high inside of the cell of solutes, then we're going to have water move into the cell and it prevents it from being lost in the environment. All right, so we're going to go ahead and move on to the next part in the interest of time. You guys can keep the questions coming, and if I don't have time to answer them right now, I will definitely try to address them towards the end. All right, so the last part, question D, marine mussel larvae are evenly dispersed throughout the study area by water movement. As larvae mature, they attach to the rocks in the water. Explain the differences in the lap allele frequency among adult mussels at this sample sites, despite the dispersal of larvae throughout the entire study area. So mussels with the lap allele are more likely to survive in which environment? So if they have the lap allele, we see according to the information they gave us, what is going to be the place where they can survive best. Yeah, so they're gonna basically survive better where there's a higher concentration of salinity, okay? So mussels with LAP of 94 allele are more likely to survive in high salinity, less likely to survive in low salinity. And if you ever say the opposite, like vice versa, it's totally also acceptable. So you would get a point regardless if you say mussels without the LAP allele are less likely to survive in high salinity, and so on. Okay, so let's move on to the prediction. Predict the likely effect on distribution of mussels in Long Island Sound if the LAP 94 allele was found in all of the mussels in the population. So if all of the mussels have the LAP allele, they're basically going to be able to have more of them in a high salinity environment, right? So we're going to see the mussel population increase in high salinity. Same thing if you said the mussel population will decline in low salinity. That's also a very accurate prediction. And the last part of the question, justify your prediction. Mussels in high salinity with LAP94 allele will osmoregulate. That's kind of, I don't know if a student would actually say that for like word for word, but if you said mussels in high salinity with the LAP allele will be able to control their environment's salt concentration, that would be acceptable as well. All right, so what questions do you guys have before we move on? Any questions before we move on to the next question? All right, you guys have done an awesome job. Thank you so much for participating. If you're just joining us, make sure you pull up the FRQ questions for 2016 so you can follow along on a piece of paper. We are moving on to question 2, which is another 22-minute question, but we are only going to look at part C and D for today because the other parts are not relevant to the test that we're taking this year. So again, I'm going to suggest that you read the question carefully and then break it down. So let's look at the question. It starts off by showing us a graph, okay? And then we're going to basically say, okay, the first thing, we're not gonna analyze the graph just yet, but like if you have a different way of doing this, different than I obviously do, you can start by analyzing the graph. So feel free, you know, it's your choice, but I'm just giving suggestions as to what I think would be the best thing. So let's just read what it says. Bacteria can be cultured in media with carefully controlled nutrient composition. The graph above shows the growth of bacterial population in a medium with limiting amounts of 2 nutrients, 1 and 2. All right, so now we're skipping A because it's asking us about population density, and that's not something that's going to be covered on this year's test. It's also asking us about growth rate, and so we're skipping it. All right, so part C, identify the preferred nutrient source of bacteria in the culture over the course of the experiment. Use the graph to justify your response and propose one advantage of the nutrient preference for an individual bacterium. So now we have to read the graph, okay? Now I'm gonna say, okay, let me look at this question 2 graph. I see, oh, I'm sorry, I skipped my own step, which is to summarize for you guys. But basically, bacteria can be grown using 2 nutrients is what it told us so far. Okay, so now we look at the part C. We're gonna look at the graph. What do we see on the x-axis? And what about the y-axis? What is happening to the bacterial population over time? Wait, was the graph on the first page only for questions? Okay, yes, Philly, just to answer your question real quick, the graph was for only for that question, so we're moving down this to the next part. Okay, yeah, so it's increasing, right? Bacteria level is increasing, the population of bacteria is increasing. As you guys may remember, bacteria, they basically grow by dividing, and so they grow exponentially, they grow very quickly. And so we're going to look at, let's continue. So it's asking us, what is the preferred nutrient source? Okay, so going back to the graph, we see on the x-axis that time is going by, okay? As time goes by, the population of bacteria increases, and then we can see the bacterial population density in cells per milliliter on the left side. And then on the right side of the graph, which they normally don't do, I mean, not don't do, this is not a graph that I'm actually very familiar with, but it has relative concentration of nutrients. So there's two axes basically going on. So showing two different things at the same time. So what we can say though is if you're looking at nutrient 1, you notice nutrient 1 decreases first, right? And then as we go on, we see nutrient 2, after about 7 hours, it also decreases. Okay, so that's what you need to look at first. You want to say, okay, figure 1 is bacterial population growth in the presence of 2 nutrients. And we see the time, we see bacterial population increasing, and we see the relative concentration of the nutrients. All right, now that we analyze the graph, we can move on. So, let me go back to the question. Which nutrient is preferred? That's the first thing, is you're just identifying it. That's all you need to say. Nutrient 1, yes, that is correct. All right, that is done. We're done for that point. We got one easy point. Justifying it. So now we need to know, why is it only nutrient 1? Or why is nutrient 1 preferred? So in order to justify this, we could say nutrient 1 is preferred because that is the one that is consumed first. And if you use the graph to justify your response, you would say when both nutrients are present in the growth medium, only nutrient 1 is used. Or you can say nutrient 1 is used between hours 0 and 5, and then nutrient 2 is consumed later on. So those would all be using the graph to justify your answer. Yes. You want to try to be as specific as possible. Instead of saying in the graph you can see that it is used up faster, you might want to say in the graph you can see nutrient 1 is used first between hours 0 and 6. Okay. Let's move on. So we identified, we justified, that's 2 points. Now we need to propose an advantage. One advantage of the nutrient preference for the individual bacterium. What advantage is there to actually having this? Why do they prefer having nutrient 1 over nutrient 2? So if we said nutrient 1 is widely abundant, it's not incorrect, but it's not necessarily addressing the question. Nutrient 1 is just as abundant as nutrient 2, so that wouldn't be one of the reasons. Can anyone else think of a possibility? What would be an advantage to having the nutrient preference? So let's see. Okay, yeah. If we always can go back to energy, right? If it requires less energy for the cell, then they're gonna choose whatever requires the least amount of energy from them. So it could be possible that it's easier for the bacteria to break down nutrient 1 and use less energy doing that than nutrient 2. Okay, so you could either say the advantage is they do not spend energy making enzymes or proteins that the cell doesn't need. Maybe they do not have to spend energy making, or maybe nutrient 1 provides more energy. Okay. One of the places where we actually see this, I know this is kind of a side note, but if you remember, bacteria like to eat sugar. Okay, so bacteria, that's what they basically eat all the time. They like to eat glucose. And so when glucose isn't present, what sugar might bacteria be eating? Does anyone remember? If glucose is not present, the bacteria will eat lactose. Yes, and they're going to break that down. So that's where the lac operon comes in. If you guys remember, I think it's important, not on the test per se, but whenever you're learning new things, it's always good to make connections to things that you've learned in the past. So if you guys remember the lac operon, that is when the bacteria expresses genes to break down, expresses genes to make enzymes that break down lactose. All right, so let's move forward. All right, so now we're on to part D. Describe how nutrient 1 most likely regulates the genes for metabolism of nutrient 1 and the genes for metabolism of nutrient 2. Provide 2 reasons that the population does not grow between 5 and 6. So nutrient 1 basically promotes the expression of genes required for metabolism of nutrient 1. That would be your description. Nutrient 1 promotes expression of genes required for metabolism of nutrient 1. Just like the lac operon, if lactose is present, it's going to start signaling that we need the lactose enzymes made so that we can break down lactose. Okay, so now provide 2 reasons that the population does not grow between hours 5 and 6. This is like a big stretch for me. I wouldn't really know what to say in this situation, so this is pretty difficult. But I think the most likely prediction or reasoning that the population wouldn't grow between 5 and 6 is because nutrient 1 was completely depleted, and it takes time for the bacteria to produce the enzymes that are required to break down nutrient 2. Okay, I know that one is kind of a stretch, but like we said before, if you always just attempt the question, attempt the answers, it's better than writing nothing. Obviously, if you have to choose between something that you know really well and something you're not really getting, answer the part you know the best first and then go back to the part that you're struggling with. But yeah, I think it makes sense when you see the answers, of course, but you don't always get the answers. So let's see what some of the questions are. Okay. Yes, you can say that either one of those would have been correct. So like sometimes you might say the opposite. You might say it suppresses the genes for the expression of the enzymes that break down nutrient 2. So that's also acceptable. So whenever you do like the opposite, like it's either like you can say, oh, this is increasing, or you can say this is decreasing, and you give the reason, then yeah, it's going to count for the full point. All right, perfect. So we are going pretty good on time. So that was, both of those were 22-minute questions. Obviously, we only did half of the second one, so that's fine. But let's continue. All right, we are actually going to skip on to question 4 because question 3 is not necessarily relevant to the test. It's always nice to practice more FRQs if you have time, but in the interest of our time, we're going to move on to question 4. This is a 6-minute question. Obviously, on the test this year, you're not going to be given a 6-minute question. You're going to be given a 15-minute question. So it's probably not going to be this short, but we're still going to practice this one. because it's good review for other concepts. Let's see. Okay, question 4, go ahead and read it. We're gonna break it down. The first thing I noticed when reading this is that we are looking at a eukaryotic cell. What are some things that you remember about eukaryotes? Anybody? Type it in the chat box. All right, I'm hearing they have membrane-bound organelles. That is 100% correct. They have a nucleus, yes. Eukaryotes have a nucleus. All right, let's continue. So I'm going to just say we have a piece of DNA that is being transcribed to mRNA and translated to make a protein. All right, so let's start answering the parts. So once we read the question, we answer the parts. We are on question 4. The figure represents the process of expression of gene X in a eukaryotic cell. Part A, the primary transcript in the figure is 15 kilobases long, but mature mRNA is 7 kilobases in length. Describe the modification that most likely resulted in the 8 kilobases difference in length of the mature mRNA molecule. Identify in your response the location in the cell where the changes occur. All right, so the first thing is we need to basically try to understand what happened to the primary transcript, and we can look at the diagram at this point and we can make some observations. So first of all, the first thing I'm seeing in the diagram is the DNA has introns and exons, okay? Then I see the primary transcript that they're talking about. They're saying that one's 15 kb long. And then we can look further down on the diagram and we see mature mRNA. And now we only see the little black sections, which means all of the exons are still in the mRNA, but all of the introns were taken out, okay? So if we want to describe the modification that resulted in this difference, basically in the mature mRNA only being 7 kb long, we have to say that the introns were taken out, okay? If you remember what that's called, you can type it in the chat box. It's not quite a deletion. This is the process of transcription. Transcription is definitely happening, but we call it something specific. We can call it mRNA processing, okay? mRNA processing. You might have also heard of it as, what is it called? mRNA editing, okay? All right. You guys are cracking me up with the comments. I can't even focus over here. Okay, so let's move on. Next thing is identify in your response the location in the cell where the changes occur. So where are we looking at this primary transcript was edited before it left the certain part of the cell? So what part of the cell is DNA found in? Yay, it's found in the nucleus, right? So DNA is in the nucleus. We have transcription taking place in the nucleus, right? And that's where this modification occurred. That's where all the mRNA editing takes place. And then it goes onto the cytoplasm to meet up with the ribosome, either free-floating ribosomes in the cytosol, or it can be ribosomes found on the endoplasmic reticulum, the rough ER, okay? All right, let's go on to the next thing. So we got 2 points. We said, we described that it was removing introns, and we identified that this is happening in the nucleus. So now we can move on to our prediction. Predict the length of mature gene X mRNA if the full-length gene is introduced and expressed in prokaryotic cells. Justify your prediction. So basically, you would have to have a little bit of prior knowledge for this part. So sometimes they don't give you all of the information that you're going to need, but you could predict that the length is going to be 15 kilobytes because, and the justification for that prediction is that the mRNA processing typically does not occur in prokaryotes. So yeah, that's that. Just as a refresher, you guys should probably go through just the function of each organelle and the pathway that it would take. So you can start with like, once the proteins are made, then it's going to go for modification and so on. It can go to get packaged and sent into the cell wherever it's needed in the cell, or proteins that are going to be sent outside of the cell might be released through the cell membrane using vesicles. So you can go ahead and review all of that information when you have a chance, okay? All right, so let's go, let's move on to the next question. Okay. I need to go to the next slide, but I lost my slides. Okay. Here we go. Question 7. So this is another one. If you were in my AP Bio class, we have gone over this question before, but it doesn't hurt to go over it again. Let's say we are going to start with the steps. So step 1 is to read the question. So I want you guys to go ahead and read the question on your own, and then you can summarize it on your own. And then you can check my summary to see what you came up with. So I'm going to give you guys a few minutes, and we'll be back to discuss in just a couple minutes. Okay, so you should be reviewing question 7. If you are ahead of schedule, you can also go to any of the other questions and start practicing them. It's totally up to you. So I'm going to go ahead and read the question out loud for us. In a certain species of plants, the diploid number of chromosomes is 4, 2n equals 4. The flower color is controlled by a single gene in which green allele is dominant to the purple allele. Plant height is controlled by a different gene in which the dwarf allele is dominant to the tall allele. Individuals in the parental or P generation with the genotypes G, G, D, D, and little g, G, D, D were crossed to produce the F1 progeny. Okay, so there's a lot of words in here that maybe you do or don't know. What I would suggest is let's just look at the key things. So the first thing I noticed reading this is going to all be Mendelian genetics. So basically, you can make a key. You can write down all of the information that it gave you by making a key. So what I always do is I like to just put like, for example, I put the big G is obviously dominant. So big G would be equal green, little g equals purple, big G would equal green, big G, little g equals green, and little g equals purple. So you can do that for both alleles that they gave you. You can make a key for both of them. The second thing I'm going to do is summarize the question in my own words. So a plant has 4 chromosomes. The flower color is determined by a single gene. We already said G is dominant and it's green, to purple, which is recessive. Plant height is controlled by a different gene, as the dwarf allele is dominant to tall. So just a fun fact. Let's see. This live session is about to end in about 8 minutes, so thank you for sticking with me. This is the last question we're going to go over. Okay. So let's go ahead and go to the next question. So now we're going to answer the parts by staying organized. You have to make sure you label A. So in the question 1, it's asking us, first of all, if you didn't know, the P generation is the parental generation. The F1 generation is the first generation. They call it something else, but all you need to know is F1 means first generation. And also the fun fact I was going to share with you is that most people think that tall is dominant, but actually being short is a dominant trait, and having 2 recessive alleles for that trait would make you tall. So let's go back to the questions. Construct a diagram. We're actually going to skip question A because it's just going to take us too much time right now. But you can also remember that you can also look up AP Bio scoring guidelines for every year, and that will give you all the answers that you can check your FRQs to. So once you actually go through and you complete all the FRQs, then you can check your answers using the scoring guidelines. So that's one way you can check your answer for part A. For part B, we need to predict the possible phenotypes and their ratios in offspring of a test cross between F1 individual and a GGDD individual. So you would make, for this part B, you could potentially make one of the dihybrid crosses, and you would do, you could do the full chart, but you don't need to do that. If you actually just do the first part of the chart, you would see that you're going to get 1 green tall, 1 purple dwarf, 1 green dwarf, and 1 purple tall. If you don't remember what a dihybrid cross is, you might want to refresh on that. Also, monohybrid crosses are just the single genes that you can also do. Part C, it says, if 2 genes are genetically linked, describe how the proportions would differ from the test cross between F1 and a GG individual. So if they're linked, they're going to be on the same chromosome, okay? And it's going to say the majority would have greater than 50% would have the parental plant phenotypes. We would see greater than 25% would be green dwarf plants, and greater than 25% would be purple tall plants. You kind of have to really work through this question in order for you to keep up with all of the different parts. But like I said, in the interest of time, and also because I'm not showing you guys the diagrams, you may not be able to see this. Okay, so last thing before we end our live session, do you guys have any questions about the topic? Any clarifications that you need? Anything that you even maybe are struggling with? Any questions with what you're expected to do or how you can study for the AP test? All right. Well, since I don't see any questions, one thing I wanted to just recommend to you guys, if yes, you can always email me, Kevin, because you're my student. If you guys have other questions that come up as you're studying for the AP test, you guys can continue to look at Fiveable. Fiveable has free cram sessions available, or not free. Most people have to pay for the cram sessions. If you guys have a cram pass, you can use that. If you don't have a free cram, if you don't have the cram pass, they offer a free bio study guide on the Fiveable website, which literally lays out every single thing that you can be studying from Unit 1 all the way through Unit 6. And it gives you an amazing amount of resources. So make sure that you guys check out the AP Bio study guide on Fiveable, because that is something that is going to be super helpful, I believe. Also, we're going to be live streaming once a week for FRQ practice. So I'll be back next week to help you guys with another question set, not the FRQs from 2016, but we'll look at FRQs from another year, and we'll go through them together like we did today. So thank you guys so much for joining me, and thank you for those of you who participated. You guys did an awesome job. So I hope to see you guys next time we do a live stream together. You guys can go ahead and log out, or if you have any questions, I'll be here for another minute or two. Thank you for joining us. I don't have the exact time for the live session, Diego, but I do know it's going to be next week. I think it's going to be next Wednesday, and probably the same time. I just don't want to give you wrong information. But yeah, I hope that I'll post it on our Google Classroom, and then also on Fiveable. It also links, for those of you who are not in my class, it also links all of the information. Okay, thank you guys. Bye. Have a great day. Stay safe. Take care. Miss you guys. Oh, bye, Brisa. Bye, Kevin. Oh, you guys.